Where we left off, by about 1870, just about everybody in the scientific community who'd studied the matter accepted the idea that life on Earth had come into existence by some kind of natural process that was going on today and still could be studied. However, at best, natural selection was seen as one of many processes that might cause this change. At worst, it was maybe only a minor one, only a curiosity. And we've talked about the reasons why. Lord Kelvin had come up with what seemed like unassailable mathematical demonstration that the Earth just wasn't old enough. That somewhere between 20 and 400 million years old, probably closer to 20 than to 400, Kelvin thought. Clearly, this would just not be enough time for natural selection to go from the goo to the zoo to you, to generate all of life's diversity. Darwin had always emphasized that it was a very Lyellian, slow and gradual process. In fact, Darwin somewhere said, "I always felt as if my books came half out of Lyell's brain." So, so it doesn't look like there's enough time. Fleming-Jenkin argues that favorable variants couldn't be selected for because they would have to interbreed with individuals that didn't have those. That's our mutant long-necked giraffe who's only got short-necked giraffes to mate with. And that ends up diluting out or washing out variation in the population. There's also the fact that neither Darwin nor, to be fair, anybody else really understood where variation came from or how it can be inherited. And you have to remember when you're studying this period that Mendel is still this slightly eccentric monk raising a bunch of peas in the greenhouse at the Abbey of St. Thomas in Brno, in what was then the Austro-Hungarian Empire. Nobody else really has any idea of how genetics operates. And finally, natural selection just didn't seem like it was a good scientific law, and it didn't seem like it had any predictive value to it. As one detractor called it, it was the law of higgledy-piggledy. So yeah, but dun dun dun. Kelvin's objection started getting put away in 1895 when Henri Becquerel and then the Curies who followed him discovered this strange new phenomenon called radioactivity. So here you've got Marie and Pierre Curie who have just gone through 20 tons of uranium ore in order to isolate a tiny lump of an undiscovered chemical element called radium. Radium, which is giving off some sort of energy so enthusiastically that radium glows. They don't really do this now, but they used to make glow-in-the-dark watch faces by painting the dials and the numbers with radium paint. Not so much now because there's a radiation hazard, not so much if you wear a watch, but if you're one of the people making the watches, that tends to be quite dangerous. But that was discovered starting in 1895. And ultimately, that's the answer to Kelvin's objection, because the Earth isn't cooling down the way Kelvin thought it is. Kelvin got his numbers right, but what he didn't know was that the Earth has an internal heat source, something on the order of 3/4 of the heat that naturally exists in the Earth comes from the breakdown of naturally occurring radioactive elements. So that's why the Earth is not cooling down or at least not nearly as rapidly as Kelvin thought that it was. We've got our own little internal furnace, as it were. And I'm not going to have time to go into detail as to how exactly we do this, but you can take certain rocks. Not all rocks will this work for, but many igneous rocks it works. Igneous rocks are rocks that used to be molten, like volcanic ash and lava. You can break one of those rocks down and measure the amount of a radioactive element and the amount of the daughter element, the element that is produced by radioactive decay, and use some basic physics and basic algebra and get an estimate for the age of the rocks. That's what this gentleman is doing here. So the older a rock is, the more daughter element builds up and the larger the ratio of the daughter element to the parent element. So for example, if you've got a rock that contains uranium, well, uranium decays into lead. To be a little more specific, uranium 238 decays into lead 206, I think, and uranium 235 decays into lead 207. And this happens at a known rate. It's not a linear rate, it's exponential decay, such that after a certain period of time, half the uranium's gone, and after a second interval of time, half of what's left is gone, and after a third interval of time, half of that remainder is gone, and so on. And the calculation is pretty easy if you know the half-life and you can measure the parent and the daughter elements. And there's various ways that we don't have time to get into as to how you can cross-check the reliability of one of these radiometric dates. And by the way, one type of dating that uses radioactivity is carbon dating. Carbon dating is only accurate to about 50,000 years ago. The vast majority of fossils, you can't get meaningful carbon dates, although you can do this with a lot of human artifacts. For most rocks, you'd use elements with a much longer half-life, like potassium decaying to argon, uranium decaying to lead, occasionally something more exotic, like samarium decaying to neodymium or something like that, but that's kind of obscure. We now know that the oldest rocks in the solar system punch their clock at 4.6 billion years old. The oldest solid rocks that you could actually walk over on the surface of the Earth are about 4 billion years old. Life appears very soon after that. Safe to say that by about 3.8 billion years old, we've got microbial life. And the oldest complex fossil animals is something of a judgment call as to what you're going to call an animal fossil. Far as I'm concerned, probably at about 700 million years old. So the Earth is far longer than Kelvin could have imagined, because Kelvin didn't know about radioactivity. By the way, Arthur Holmes was still an undergrad at the University of Durham in northern England when he tried to date rocks this way, and he basically got the right answers. The oldest rocks he could find to test turned out to be about 1.6 billion years old. We have since found even older rocks, specifically a rock layer called the Acasta gneiss up in northern Canada. And the oldest solid rocks that you could, walk over and climb up on the Earth's surface are at about 4 billion. The age of the Earth is known to be about 4.6. The reason for the discrepancy is the earliest Earth was molten and didn't have solid rock on it at all. So much for Kelvin. Kelvin, by the way, was very old when radioactivity was discovered, and he thought it was a hoax. Uh, as he got old, he started making lots of authoritative pronouncements like heavier than air flight was impossible, and X-rays were a hoax, and the world was going to end because all of the plants on Earth were only capable of producing about half of the oxygen that animal life needed, which, by the way, is true, except he forgot or didn't know about microscopic algae in the oceans, which produce the other half of the world's oxygen. And I think I mentioned this in marine bio. Anyway, as for Jenkins' blending inheritance, that's Gregor Mendel. And there's you a little quote from a paper that he put out in 1865 called Experiments in Plant Hybridization. Nobody paid much attention at the time because Mendel published his work in the Journal of the Natural History Society of Brno. Brno is the name of the city in the Czech Republic, spelled Brno, where Mendel lived and worked, and not a lot of people read journals from Brno. He did send copies to other scientists. The story goes that he sent a copy to Darwin, and Darwin didn't read it because Darwin was not really good at foreign languages, and Darwin could only read German very slowly and rather painfully with the help of a dictionary. And as far as we can tell, he never read Mendel's paper, or at least that is the story that goes around. Mendel's work knocks Fleming-Jenkin out of the park ultimately because inheritance is not like mixing paint. Remember, I compared blending inheritance to being like, imagine you've got a bucket of water and you add 1 drop of food coloring. The food coloring spreads out and it gets diluted with time until eventually you have a bucket of clear water that might be the very tiniest bit of pink, but you can't even see it, and you'll never get that droplet back, right? No matter how much you stir that water, you'll never recover the droplet of food coloring. Mendel was the first to realize that inheritance is particulate. I should go back one. That, yeah, inheritance is a result of, as he put it, constant characters in all the associations which are possible according to the laws of combination. He's talking about genes. And he didn't know at the time what genes were. And this is a little oversimplified, but you can think of genes as being like these little particles rattling around inside cells. You know, pea plant cells have some little thingies in them that can be either capital P or lowercase p, and they determine whether the flower is purple or white. I'm sure please tell me you remember all of this. And so instead of mixing, food coloring into water, it's more like mixing marbles. If you add a red marble to a bucket of white marbles, the red marble doesn't disappear. It's still there. It's not lost. And new alleles, new genetic variants, if they're recessive, they might not be obvious, but they're not diluted out. They're still present. They may become more common. They may end up showing up in the phenotype. So much for Jenkin. Now, Lamarck, as I mentioned, was really big at this time. Probably the biggest anti-Lamarckian was a German named August Weismann, who argued against neo-Lamarckianism. He was mostly a theoretical biologist because he had very poor eyesight and couldn't use a microscope effectively. But he hypothesized that there must be in a living body or a living cell some kind of physical stuff that's responsible for heredity. Um, that heredity must work because there's some substance that somehow carries hereditary information. And he called this germplasm. We would now, of course, say, oh, this is DNA. Uh, I guess DNA was known at about this time, but nobody knew what it did. But Weissmann turned out to be quite right. And his theory went on, he went on to propose something called the continuity of germplasm. And the idea there is that whatever this hereditary stuff is, it's set apart from the rest of the body. In your case, it would be the DNA in your egg cells or sperm cells, whichever the case might be. And there's no way, Weissmann thought, that any kind of signal from the rest of your body can be transmitted to alter the germplasm in your eggs or sperm. You can body build all you want to, but there's no way that your bulging muscles can reprogram the DNA in your eggs or sperm, which means that even if you bulk up to look like Jason Momoa, your kid is not going to be born looking like Jason Momoa, which is probably just as well. The guy's a pretty handsome man, but a baby that looked like that would be a little scary. But yeah, there's no way that Jason Momoa's giant muscles can reprogram his DNA. That isolation of the DNA that gets passed on is still called the Weissmann barrier. And I mention that because it still comes up in discussions of bioethics. Take gene therapy. There are ways, and they sometimes work, that you can take a person with a genetic disease and insert genes that correct the disease, genes that correct the deficiency, gene therapy. Bioethics folks go back and forth over, what are the ethics of this? And the current consensus is it's okay to engineer the DNA in the rest of your body if you need to cure a disease, but it's not okay to engineer the DNA that a person is going to pass on. So if you have doesn't even have to be genetic diseases. Let's say we find a gene that makes you look like Jason Momoa, assuming you want to look like Jason Momoa. It might be ethical to insert that gene into the genome of your body so that you get big muscles and you look like Aquaman. But it would not be ethical to insert that gene into your eggs or sperm. Would not be ethical to make a permanently hereditary change in your DNA. That's called crossing the Weissmann barrier. So I'm not taking a formal position on this, but if you ever read up on the bioethics of genetic engineering, you'll see that phrase, the Weissmann barrier, mentioned. And this is the guy it's named for. Weissmann was shown to be pretty much right in 1873 when a dude named Schneider made these drawings of cells in the tail of I believe it's the tail of a larval salamander. And on the left, there's an ordinary cell. And then going from left to right, you can see some drawings that ought to look painfully familiar from Bio 1440, because the second from the left, you can see that's a cell and it's got some kind of wiggly things lined up in the middle. And then on the third one, you can see those wiggly things are pulling apart. And there you can see they have pulled apart and the cell is dividing over there on the right. Anybody can recognize these as, metaphase, anaphase, and telophase of meiosis. So Schneider's discovered these little stringy things, which he calls chromosomes. And Weissman concluded that chromosomes were this germplasm, that there is a hereditary substance, a material bearer of hereditary tendencies in the nucleus of the germ cells, in that part of it which forms the nuclear thread. He's talking about DNA making up chromosomes, and he basically got that right. One more piece of the puzzle before we get to the modern synthesis. Here's a Dutch guy named Hugo de Vries who starts experimenting with crossbreeding plants, and without realizing it, he replicated some of Mendel's work, except he didn't work with peas. He worked with a rather attractive garden flower called the evening primrose. This is the evening primrose, ironically named after Lamarck. This is Oenothera lamarckiana. And animal breeders and plant breeders had noticed this sort of thing in the past. They used to call them sports, sports of nature. Sports in the sense of games. It's like nature is playing a game with you. If you're breeding your cows or your cats or your cabbages or what have you, and one member of the population has some kind of trait that you've never seen before. They used to call that sports of nature. De Vries noticed this too, and he ended up calling these mutations. He's the guy who coined that word, these sudden changes in color or in shape from one generation to the next. And on the basis of his observation of mutations, he argued that traits must be inherited as units rather than blends. So he had rediscovered Mendel's finding that inheritance is particulate, that there's these little particle thingies that are responsible for traits. Mendel had just called them elements, elementen. De Vries called them intracellular pangenes, which later got whittled down just to genes. And then, within a few months, in the first half of 1900, Hugo de Vries happened to find a dusty old copy of Mendel's paper and realize that he was repeating Mendel's work and Mendel had gotten farther with it. And two other scientists independently discovered Mendel's work at almost exactly the same time, Karl Correns and Hugo Czermak. And that was the last missing piece. The idea that genes are these little particulate things and they independently assort was ultimately what knocked out blending inheritance. Our giraffe who's suddenly shows up with a long neck, he might only mate with short-necked giraffes, and the babies might be short-necked, but his long-necked genes are going to survive, and eventually they're going to come back together. The genes for a long neck aren't lost. They're not diluted like food coloring in water. They may be masked, but they can come back like red marbles in a jar of transparent marbles. There was a very long debate as to just how important this was, by the way. There was a whole school of thought called the biometricians who argued that genes might be interesting, but they weren't really important because most traits don't follow Mendelian rules. By which I mean things like, height, weight, physical proportions, skin color, things like that. Those don't come in a nice, neat, small set of traits. Pea flowers are either purple or they're white. Human skin is not either black or white. We're not really ebony or ivory. We range from a very light peaches and cream all the way to a rich ebony, and we've got every shade in between. We don't really fit into neat classes like that. Pea plants can be either tall or dwarf, one or the other, but we're not all either NBA centers or Kentucky Derby jockeys. We have a few people that are crazy tall and a few people that are really short, and most of us are somewhere in between on a continuous range of variation. And at first, it was not clear how you could use Mendelian genetics to explain something like that. So the biometricians are arguing that Mendel's work might be interesting, but it doesn't really explain the most important things that you'd want to know. There was a long debate about that for a while, and it finally got cleared up by about 1925. And we'll talk later, sometime next week, hopefully, as to how that debate was ultimately resolved. So De Vries explains mutations as changes in these genes in this book that he wrote called Die Mutationstheorie. He was not, by the way, a supporter of natural selection. Remember, I mentioned that Huxley had thought that change might be rapid, that you could get major change in phenotype within just a single generation. And De Vries went along with this. He was a saltationist, thought that new species might spontaneously appear in one generation by mutation. We'll later see he wasn't entirely wrong. There are actually some ways in which new species can appear surprisingly fast. And as I've mentioned, you have this argument in the early 1900s between the Mendelians, who attributed everything to these isolated, discrete genes, and the biometricians, who emphasized the inheritance of continuous traits. There were some Mendelians who kind of overstated things. A guy named Goddard, who I think might be mentioned in your reading, a big-time proponent of eugenics, argued that what he called feeble-mindedness was inherited as a single Mendelian trait with two alleles. It isn't, by the way. There are some genetic reasons why you can be developmentally delayed, but they don't boil down to one single gene with two alleles. Human intelligence is much more complicated than flower color in pea plants. So there were people on, there are arguments to be made on both sides. So here's a claim that the Mendelian army has turned the flanks and pierced the center of the biometrical army. And if we skip over a great deal of complex controversies in history, by about 1930, people like the American geneticist Sewell Wright have welded together Mendelian genetics theory and Darwinian natural selection and produced a powerful body of work that we now often call neo-Darwinism. Or is also often called the modern synthesis. Now, the modern synthesis is not everything there is to know about evolutionary biology. There's a lot more to it than that. But this is still kind of the basis for what I think of as, classic evolutionary biology. It's still, a very important underpinning of the science as we understand it now. Whew, oofta. Okay. For purposes of this course, I am going to assume that you remember the foundations of Mendelian genetics. Genes exist. We can, for right now, we can kind of pretend that genes are little particles. Of course, they're not. They are lengths of DNA, and they can be subject to everything from transposons to alternate RNA splicing to RNA interference and who knows what. for what we need to do here, we can kind of pretend that our genes are, little colored ping pong balls or something like that, and that genes exist in one or more variant forms called alleles. And one allele of a gene may be dominant or recessive to any of the others. And of course, you can have codominance, as in human blood groups, or incomplete dominance, as you have in, I don't know, roan cattle and pink snapdragons and things like that. If you have forgotten all of your Mendelian genetics, please go back and take that out behind the woodshed and dust off your old notes and give yourself a quick refresher in it. When I started teaching this course, couldn't have been that long ago, 23 years ago. Oh my God, I can finally say this. All right. I've been teaching evolution since before some of you were alive. There, that felt good. I ran into some problems because people used to take the genetics course and then do a complete and total memory dump. And I'd have to go back and explain what alleles were. That hasn't been so much of a problem in recent years. But if you're fuzzy on it, make sure you get yourself up to speed. I haven't checked, but you are technically supposed to have taken genetics as a prereq for this course. So let's say we have a population. You can define a population as just a group of individuals of a particular species living in a particular area and potentially all capable of interbreeding with each other. That'll do. And within that population, you have alleles, E-I-E-I-O. Oh, hey, let's see. Sorry, I've got some cats in this room. Oh, here's a good one. In fact, this is one we're going to use in a lab that we're going to do this coming Thursday after the first exam. If you look at cats, they have a gene for white spotting, and spotting is dominant to non-spotting. So if you have a cat that has the genotype that's homozygous recessive, let's call it little s, the cat will not have white spots. If you have a cat that is homozygous dominant, which we could write big S if you wanted to, the cat will have lots of white spots. In fact, so many that it may look like a mostly white cat with maybe just some pigment on the ears and the tail or something like that. It'll basically have so many white spots that the spots run together and it's mostly a white cat. If you have a cat that is heterozygous, you have a cat with small amounts of white spotting, with possibly just little white star on its face or white paws, or like Jackwagon right here, one of the cats that's asleep in front of me. He's what they call a tuxedo cat, right? Because he's got markings that make him look like he's wearing a tux. So I have 3 cats in this room right now, 2 of them asleep and one of them still got the zoomies. Fern does not have any white spots. Well, first of all, I've got 3 cats. They're all diploid. So we have 6 alleles, right? Every cat has 2 copies of each gene. So we have 6 copies of the spotted allele. Fern does not have spots, so we score her genotype as little s. Jack is heterozygous because he's got white spotting, but not very much of it. And Tsubaki is actually kind of hard to tell because she's got a Siamese pattern on top of that. But I'm going to say she probably has 2 copies of the spotted allele. I think she is homozygous for it. So we have 6 alleles. One cat's homozygous for the spotted allele, one cat's heterozygous, and one cat is homozygous recessive. So that means the frequency of the spotted allele is 50%, right? If we have one cat that's little s, one that's big S, and one that's big S, little s, well, that's 6 S's, 6 copies of the spotted allele. 3 of them are dominant, 3 of them are recessive. So the frequency of the spotted allele in the population of cats in this room is 50%. That's how you calculate allele frequencies, and we'll do some slightly more complicated examples coming up. If you have a gene that has just 2 alleles, we represent the frequencies of those alleles as lowercase p and lowercase q. Genes, of course, can have more than 2 alleles. If we have just 3, we could probably get away with calling them P, Q, and R. The pros would start representing them with subscripts, P1, P2, P3, P4, however many you need. But those are frequencies of the allele in a population. And modern synthesis evolutionary theory starts by looking at whether and how allele frequencies change. Because of selection or sometimes because of other factors, an allele that used to be rare can become more common in later generations, or a common allele can end up more rare. One case that is kind of in pop culture these days is that allele frequencies by themselves can sometimes be used to indicate the population's past history. And you can learn some things about, I guess, any population, but the one that tends to grab the spotlight these days is human history. You can look at a human population's allele frequencies and gain clues as to the past history of that human population. The alleles that's easiest to do with are alleles that only appear in a single copy per person. And alleles that only appear in one copy per person are known as haplotypes. These are alleles that appear on mitochondrial genes and on Y chromosomes. If you have a Y chromosome, you only got it from your dad, right? You did not inherit a Y chromosome from your mom, because if you did, that would make your mom your dad, right? So a man will pass on his Y chromosome to all of his sons and none of his daughters. And so, guys, our Y chromosomes are identical to our father's Y chromosomes, except for the occasional mutation. Okay, you didn't inherit a Y chromosome from both parents and then recombine it. The Y chromosome doesn't normally recombine. Mitochondria, if you remember, right, they're the powerhouse of the cell. I think I'm supposed to say that. Mitochondria have their own DNA, and you got all of your mitochondria exclusively from your mom. Your dad's mitochondria do not get to pass their DNA on. Their usefulness ends at fertilization. And every mitochondrion in all of your cells is ultimately descended from your mom's mitochondria, and hers were descended from her mother's mitochondria, and so on. So mitochondrial DNA is only inherited from one parent. Your mitochondrial DNA you got from your mother's mother, mother. And if you're a dude, your Y chromosome DNA you inherited from your father's father. And different alleles, we don't all have the same mitochondrial or Y chromosome DNA, because those can mutate like any DNA can mutate. And variant alleles on mitochondrial or Y chromosome DNA are called haplotypes. Remember, related to the word haploid, if you're haploid, you only have one copy of each chromosome. Well, haplotypes, like haploid chromosomes, exist only in single copies. And this is actually going to be a pretty good spot to take a break. I'll come and talk more about haplotypes and their use in evolution. I've got 9:49. So let's go ahead and take a break and we can come back at 10:05. So I'm going to stop the recording now.